Chapter 11, Part 5, we're moving on to organization of nervous tissue. So again, I kind of already mentioned this. Gray matter is made up of axons that do not have any myelination. White matter, axons that do have myelination. In the brain, the gray matter is the outer cortex, and white matter is deep in the cortex, and we'll look at that when we look at the brain. When we get to that. The term nuclei, in reference to the nervous system, is referencing clusters of gray matter deep within the white matter of the brain. In the spinal cord, it's the opposite. The white matter is the outer layer, and the gray is deeper. And the white matter of the CNS forms nerve tracts, or basically, I think of it as nerve roads. So it's tracts of nerves or groupings of nerves that go from one place to another. Peripheral nervous system gray matter is groups of cell bodies called ganglia. So electrical signals. Cells produce electrical signals called action potentials. And it's a transfer of information from one part of the body to another. Notice how I have turned this bottom part of the slide very big, purple, some of it's bolded and underlined. What causes a membrane potential? A membrane potential results from an ionic concentration difference across the plasma membrane, as well as the permeability of the membrane. So a difference in specific ions, whether it be sodium, potassium, chloride, negatively charged proteins, and so on, those allow for a membrane potential. If everything is exactly how positioned perfectly in order to allow for a electrical signal to travel. So we're going to talk more about this as we go. So ionic concentration differences across the plasma membrane. So there's a lot of this section of the chapter that it's just remembering facts that will influence your understanding of an action potential, if that makes any sense. I hope it does. So if you know all these little individual parts and then you kind of put it all together when we're talking about an action potential, it might make more sense. So the ion concentrations that are found across that plasma membrane are a result of two processes, the sodium-potassium pump and that membrane permeability that was mentioned already. So to start, there are high concentrations of sodium and chloride outside of the cell. And I'm really going to focus on sodium and potassium here, okay? I mean, it's important to know there's other stuff, but you should know that we're really focused on sodium and potassium here. So high concentrations of sodium and chloride outside of the cell, but I want to point out that there's some potassium outside of the cell as well. Inside of the cell, there are high concentrations of potassium and proteins. And these proteins are actually negatively charged, but we'll get to that as we go. So very high sodium outside, very high potassium inside. And the interesting thing is here is that these are both positively charged. And these are the things that we mentioned, we talked about a little bit when we talked about muscles. So these are the things that are moving back and forth across the cell membrane that changes that the ability for things, or increases the ability for a electrical charge to travel. So a steep concentration gradient or difference of sodium and potassium, but in opposite directions. So permeability characteristics of the plasma membrane. So proteins are synthesized in the cell. They're large and negatively charged, and they cannot go through the plasma membrane. So proteins are going to stay put, okay? And notice that proteins are inside the cell. So those things are gonna, for the most part, stay inside the cell. Chloride is repelled by proteins, and they get away from the proteins through open channels that are there specifically for chloride. So they're going to go on the opposite side of that cell membrane. So there are gated ion channels that open and close in response to a stimulus, and when they open, they change the permeability of the cell membrane. There are leak channels, and again, we've talked about the different types of channels, and so we're revisiting them. Leak channels are basically always open, and they are partly responsible, not completely, but partly responsible for the permeability when the membrane is at rest. Leak channels are specific for one type of ion, for the most part, and there are many more leak channels for potassium and chloride than for sodium. So at rest, there's more potassium and chloride that are moving back and forth when the cell membrane, when the neuron is polarized and ready to go. So there's more potassium and chloride moving than sodium. There are also gated ion channels, and these open and close because of some sort of stimulus. And when they open, they change the permeability of the cell membrane, meaning when they open, stuff is going to go through. That changes the permeability. It is not permeable when the gate is closed. It becomes more permeable when the gate is opened. So there are ligand-gated channels. These open or close in response to a ligand, such as a neurotransmitter that binds with one of the proteins on the cell membrane. And a ligand is, think of a ligand as any sort of chemical. It can be a hormone, it can be a neurotransmitter. And the example that we've already experienced through the muscles is the acetylcholine binding to the acetylcholine receptor on a sodium channel. And when that acetylcholine binds to that protein receptor, the gate opens and sodium is then free to travel through. The acetylcholine is like a key to open the gate. We also have voltage-gated ion channels, which will open or close in response to small voltage changes in that membrane. So at rest, the membrane is negative on the inside relative to the outside. So it's positive on the outside, negative inside. When that cell is stimulated, the relative charge changes because the voltage-gated ion channels will either open or close. And the most common voltage-gated channels are sodium and potassium. These two ions are the two that we're going to really focus on, that we've talked about in the muscle system, that we're going to talk about in this system, as far as being able to transmit signals or carry that signal via the way they open and close. In cardiac and smooth muscle, potassium channels are important. I'm not going to talk a whole lot about this because we don't really focus on cardiac and smooth muscle. We're only focusing on action potentials in skeletal muscle for this unit. There are other gated ion channels as well. Touch receptors respond to mechanical stimulation. There's also temperature receptors respond to temperature changes in the skin. So I'm going to keep going here. So establishing the resting membrane potential. So intracellular fluid and extracellular fluid are electrically neutral, but there's a charge across the membrane due to those anions and cations, which are sodium, potassium, chloride, the negatively charged proteins, and so on. Opposite charges across the membrane, so the membrane is polarized. So as we said before, we said that, one side, the outside of the cell has more sodium, and the inside of the cell has more potassium, and one side is positive and one side is negative. So that's referred to as a potential difference. It's an unequal distribution of charge existing between the immediate inside and immediate outside of the plasma membrane. And that potential difference is around negative 80 millivolts. Some books say negative 70, some books say negative 80, some books say between negative 70 and negative 90. I just try to make it easy and say around negative 80. It's somewhere in that vicinity. So the resting membrane potential exists in an unstimulated cell due to permeability of the membrane and differences in the ion concentrations on each side. How does that resting membrane potential exist? It exists due to these two things. And I would guess that this is going to be a test question. The permeability of the membrane as well as differences in ion concentrations on both sides. That's how we get the resting membrane potential. So membranes are more permeable to potassium due to the increased leak channels of potassium, or for potassium rather, and potassium will then diffuse from the inside to the outside of the cell. So what happens there is that there's positive charges that accumulate on the outside of the membrane. The negatively charged proteins that are in there that I mentioned before, those guys are really big. They're not going to fit through any of the channels. They're stuck. They're staying put. So they can't diffuse with the potassium. So as the potassium diffuses out of the cell, the inside of the membrane will become more negative. So sodium, chloride, and calcium do not have huge effects on resting membrane potential, since there's not nearly as many leak channels for those. So again, there's way more leak channels for potassium than there are anything else. So a resting membrane potential is mostly maintained by the leak channels and the sodium-potassium pump. With the sodium-potassium pump, we talked about this at the beginning of the semester, that for each ATP that is consumed, the sodium-potassium pump is able to pump 3 sodium ions out of the cell and bring 2 potassium ions in, leaving the outside of the plasma membrane slightly positive at rest. So this is kind of showing you an illustration of, establishing that resting membrane potential, showing that there is more sodium on the outside in pink, there's more potassium on the inside in purple. We have these negatively charged proteins, and we have, actually they're not showing any chloride there, but that doesn't matter. All right, so we're going to stop there, and we're going to move on to what happens when a stimulus comes in and depolarizes the cell membrane.